Good morning, everyone. Welcome to the class of biology on the fascinating world of biology. Throughout this course, we will learn about digestion of different foods, and food included in carbohydrates, fats, starch, glycogens, and its absorption. Here our main purpose is to improve our reading skills. Those learners which are weak in reading, here will be improve your reading skills. Now let's start to the topic digestion of different foods. Carbohydrates digestion. The food we eat many contain starch and other carbohydrates such as lactose, sucrose, and cellulose. Starch is digested by amylase. Starch digestion begins in the mouth, as food does not remain long in the mouth. Only a little starch is digested by salivary amylase to maltose. No digestion of carbohydrates occurs in the stomach. When carbohydrates enter the small intestine, they are digested as follows. Look at this graph and try to understand the process of digestion of different foods. Starch digested into maltose in the presence of pancreatic amylase, and maltose is converted into glucose in the presence of enzyme maltase. Lactose converted into glucose and galactose in the presence of enzyme lactase. Sucrose converted into glucose and fructose in the presence of saccharase enzyme. This process takes place in the small intestine. Look here and try to understand the process of conversion of starch, lactose, sucrose in different products in the presence of enzymes. Thus, the end product of carbohydrate digestions are simple sugars, glucose, fructose, galactose, which can be absorbed. Cellulose is not digested in the gut. Cellulose is not digested in the gut. Digestion of protein digestion. Proteins are digested by protease. Protease enzyme is involved in the breakdown of protein. Some protein digestion begins in the mouth, where pepsin digests protein to polypeptides. The undigested proteins which enter the small intestine are digested by trypsin to polypeptides. The enzyme trypsin is produced as inactive trypsinogen in the pancreatic juice. In the intestine, trypsinogen is converted to the active trypsin by intestinal enterokinase. Enterokinase. The polypeptides produced as are further digested to amino acids by the enzyme trypsin in the intestinal juice. The end products of protein digestion are thus amino acids which can be absorbed. Look at this diagram and chart and try to the process of digestions of different enzymes. Trypsinogen converted into trypsins in the presence of enzyme enterokinase, and proteins are converted into polypeptides in the presence of enzyme trypsin, and polypeptides converted into amino acids in the presence of ripsin enzyme. The whole process takes place in the small intestine. A cellulose diet. Humans do not digest cellulose because we do not produce the enzyme needed to digest it. Cellulose digestion is important to herbivores as they survive by eating plants. Cows, for example, consume vast amounts of grass which consist mainly of cellulose. Actually, cows do not digest cellulose themselves. Cellulose is digested for them by microorganisms which live in their stomachs. These microorganisms break down the cellulose to substance that can be absorbed by the cows. Cellulose digestion microorganisms live in the cecum and appendix of other herbivorous animals such as a rabbit. Food digestion. Food digestion. In the small intestine, bile salts emulsify fats. They lower the surface tension of the fats. That is, they reduce the attractive forces between the fat molecules. This causes the fats to break into tiny fat droplets suspended in water, forming an emulsion. Note that this is just a physical breakup, but no chemical digestion of fat molecules has occurred. Emulsification increases the surface area to volume ratio of the fats, speeding up their digestion by lipase. Emulsified fats are digested by lipase, pancreatic and intestinal lipase to fatty acids and glycerol. Thus, the end products of fat digestions are fatty acids and glycerol. Big fat. Look at this diagram and try to the process of fat digestion. Big fat drop. A big fat drop plus bile salts combine to form reaction takes place and break down the big fat drops into tiny fat droplets. Big drop plus bile salts and converted the big drop into tiny fat or small fat droplets. Bile salts emulsify fats into tiny fat droplets. Physical digestion is the mechanical breakdown of food into smaller particles. Physical digestion increases the surface area to volume ratio of the food so that digestive enzyme can act on it more efficiently. Chemical digestion refers to the hydrolysis of large food substance by digestive enzymes to small soluble molecules. In the mouth, food is broken up by chewing and the action of salivary amylase. Food passes down the esophagus by peristalsis. Look at this chart and try to understand the region of digestion, secretion, source, enzyme, and action. Region of digestion, secretion, source, enzyme, action. Mouth, saliva, salivary glands, salivary amylase, pepsin, converted a starch into maltose, protein into polypeptides. Stomach, gastric juice, gastric gland, renin, soluble, insoluble, carcinogens, casein, bile, liver, renin, bile salts emulsify fats, amylase, starch into maltose, pancreatic juice, pancreas, trypsin convert proteins into polypeptides, lipase, fats converted into fatty acid and glycerol, small intestine, enterokinase, trypsinogen converted into trypsin, maltase enzyme, maltose converted into glucose, intestinal juice, intestinal glands, lactase enzyme, lactose converted into glucose and galactose, sucrase enzyme, sucrose converted into glucose and fructose, ripsin enzyme converted polypeptides into amino acids, lipase fats converted into fatty acids and glycerols. Look at this whole chart and try to the whole process of digestion, region of digestion, secretion, source, enzymes and action. Which enzymes are converted the different sugars into their products. Now we discuss the topic of absorption. Absorption. Products of digestion such as simple sugars, amino acids, fatty acids, and glycerols and absorbed throughout the small intestine, especially the ileum. The absorbed nutrients pass from the small intestine into the bloodstream. Water is also absorbed in the small intestine. Food absorption and water absorption takes place in small intestine. How is the small intestine adapted for absorption? The rate of absorption of digested food substances depends on the following factors. The surface area of the small intestine have how large the absorbing surface is. The membrane that separates the food substances from the blood capillaries. Digested food substances take a shorter time to pass through a thin membrane than through a thick one. The concentrations of gradients. The small intestine is well adapted for the absorption of digested food substances. The surface area of the small intestine is increased for absorption in the following ways. The inner walls of the small intestine have numerous folds. The inner walls of the small intestine are also lined with numerous minute finger-like projections called villi. Singular villi. These villi further increase the surface area for absorption. Under the microscope, we see that the epithelial cells of the villi in turn have numerous microvilli to increase surface area. The villi have thin walls or membranes. Epithelium is only one cell thick. The small intestine is long to provide sufficient time for absorption. The intestinal wall and the villi have many capillaries to carry away the absorbed food substances. In each villus is a lacteal lymphatic capillary surrounded by blood capillaries. The lymphatic capillaries of the villi transport fats while the blood capillaries transport sugars and amino acids away from the intestine. This continual transport of digested food substances maintain the concentration gradients for the absorption of digested food substances. Look here, the surface of small intestine, microscopic structure of villi, scanning electron micrograph of the small intestine showing the villi. Look here, villi are like this structure, and here's are the feathery pattern of the inner wall of the small intestine indicating its numerous folds. Look at this diagram and try to understand the small intestine with portions cut off to show folds and villi. Villi in a portion of small intestine. Look section of villus highly magnified through the electron microscopes and try to understand the structure of small intestine finger-like structure called villus. Here are folds are present and on the folds small finger-like structures called villi, and inner side of these lining longitudinal muscles and upper one is circular muscles, and here are two liver, and here is present intestinal gland, intestinal juice, blood capillaries, lacteal lymphatic capillaries, and here are epithelial on cell thick, and the microscopic structure of epithelial has a microvilli. Here are microvilli is present and the epithelial cells, and these structures are called digested food substance diffuse into blood capillaries. Look at the structures of small portion longitudinal structure of small intestine and the complete overview of the belly and its parts, capillaries, intestinal juice and their enzymes, circular muscle and longitudinal muscle is present in the small intestine. How does absorption takes place in the intestine? Glucose and amino acids are absorbed by diffusion into the blood capillaries of the villi. Glucose and amino acids are also absorbed by active transport. This happens when there is a lower concentration of these digested food substances in the lumen of the small intestine than in the blood capillaries. Glycerols and fatty acids diffuse into the epithelium. Here they combine to form minute fat globules which enter the lacteals. Water and mineral salts are absorbed by the small intestine and the colon. Most of the water is absorbed by the ileum. Here we looked at the absorptions that takes place in small intestine and its parts. Here we also looked at the most water which are absorbed in the ileum. Ileum is the part of small intestine. What happens to undigested and unabsorbed matter? Undigested and unabsorbed matter is stored temporarily mainly in the rectum before it is discharged as feces through the anus. This removal of undigested matter from the body is called egestion or defecation. Egestion or defecation, the removal of undigested matter. Investigations how a carbohydrates absorbed in the mammalian gut show how an experiment can be set up to show how carbohydrates are absorbed in the mammalian gut. Refer to investigation, the practical book for instructions on how to carry out this investigation. Assume that the living whisking tubing represent a mammalian gut wall. Explain your results in terms of digestion and absorption of carbohydrates in the mammalian gut. Name two enzymes involved in carbohydrate digestion and state the part of the gut they would be found in. If the whisking tubing represents a portion of the mammalian gut, that would the distilled water represent? Experimental set up to show how carbohydrates are absorbed in the mammalian gut. Look at this diagram and try to the process of how carbohydrate digested in the gut of mammalian. Look at this part, cotton thread, whisking tubing. These are the whisking tube solutions of starch and glucose. Here yellow color are the solution of starch and glucose, and the outer boundary of tube is got distilled water from which test samples are taken, and the boiling tube here. Now we take the overview of this study which we discuss in this lecture. Look at this diagram of small intestine which looked through the electron microscope, and we observed the parts of the small intestine. Here we looked at small tube and bundle of folds, villi presents, and further these parts of the small intestine look in under microscopes, and here are longitudinal muscles, circular muscles, two livers, intestinal gland, intestinal juice, blood here inside the blood capillaries present, lymphatic capillaries, epithelium capillaries are present, small intestinal portions of cut off to show folds and villi. And here we look also the internal structures of the villi. And we look here that the big fat drops plus bile salts converted the a large drop of fat in the presence of bile salt converted into tiny fat droplets like big and is converted into small droplets of fats. Bile salt emulsify fats into tiny fat droplets. And here we look the diagram of different enzymes in the present converted the starch into maltose plus glucose in the present pancreatic amylase, maltase, lactose converted in the presence of lactase, glucose plus galactose, sucrose converted into glucose and fructose in the presence of sucrase. And here we discuss about the also further enzymes and their conversions. Trypsinogenes converted into trypsins in the presence of enterokinase enzyme, and protein converted into polypeptides in the trypsin, and polypeptides converted into amino acids in the presence of trypsins. The whole process takes place in the small intestine. And other further is cellulose is digested in the herbivores in the presence of bacteria. Cellulose does not digested in humans because not enzymes are present in human to digest the cellulose. In this lecture, we discuss about the digestion of different foods in animals like carbohydrate digestion, and also discuss about the absorption in the small intestine. Food and water are absorbed in small intestine. I hope this lecture is very useful for those learners which are very weak in the reading skills. Thank you so much for listening this lecture. We will meet our in next class. Thank you.